biases and my comments on why some of these topics are important for plant breeding. So today on Review of Genetics, we'll talk about Mendelian genetics, of course, and Mendel's laws, but then we'll talk about some complication to Mendelian genetics, and then we'll talk about what I've sort of listed as Morgan's laws, and we'll explain that when we get to it. It's basically, Morgan was a Drosophila geneticist who sort of pulled all of Mendel's work and work that had gone before into a theory that all of these genes that we're dealing with are located on chromosomes, and sort of came up with the concept of genes on chromosomes and linkage. Mendel was important because he was the first person to analyze patterns of inheritance, or at least the first recorded person in recorded history. And basically Mendel deduced the fundamental principles of genetics. Now you got to realize this was at a time where we knew nothing about genes, we knew nothing about inheritance. In fact, most people didn't really believe in inheritance. They believed that there were mythical factors in the environment that sort of controlled what people and animals and plants looked like and how they performed. So Mendel's work was very important. And what's equally important, as today in some of our molecular genetics topics, we start out by using phenotypes to try to get to genotypes. Well, this is just what Mendel did. He studied phenotypes, or the physical appearance of individual plants, and inferred or deduced from that what the genetic makeup of those plants must be. So how did he do it? Basically, he made some crosses and created some hybrids, and we'll talk about this in more detail, and now is where I wish I had that nice long pointer. Basically, he started with flowers of different phenotypes, either purple flowered or white flowered. He had to remove the stamens, or the anthers, from the purple type, and he then used the white type flowers as a source of pollen. And so he brought pollen from a white plant and crossed it onto a purple plant and ensured that he had made a hybrid. And for the rest of the class, we'll talk about the importance of that for plant breeding, making sure that you're dealing with parents of known origin and that, in fact, making hybrids. After he made the hybrids, let the pollinated seed mature, then he grew out the offspring, and then he just observed what happened in the offspring. And interestingly enough, he found out that when he crossed purple flowers by white flowers, all of the offspring had purple flowers. Important parts about that: the plants that he started with, he allowed to self-fertilize. So they were true breeding. They were inbred, or very homozygous in nature. He made the cross between alternate forms of a trait, purple flowered versus the white flowers, and then he allowed those self plants, those cross plants, to self again for several generations, and he looked at the results. So when he dealt with a cross involving one trait, it's called a monohybrid cross. That F1 generation, all the plants had purple flowers, but when he grew out the next generation, he found that 3/4 of them were purple and 1/4 reverted to the white flower type of one of the parents. In fact, Mendel was lucky, or selective, probably selective, because he dealt with a number of different traits. And flower color, flower position, seed color, seed shape, pod color, and stem length. All of the traits that he dealt with and reported in his original papers, we know now were controlled by single dominant genes. And so it was very easy for him to sort of pull from all of the patterns of inheritance that were around him some assumptions based on these particular crosses, that in fact the genotype of an individual differs somewhat from the phenotype, and that in fact this phenotype with 3 purple and 1 white, that you actually could have 3 different genotypes: pure purple, pure white, or plants that were apparent mixtures, but looked to be the phenotype was the same as the purple parent. From this, he deduced that genes occur in alternate versions within individuals. Of course, he didn't call these genes or alleles, but he said for each trait that controlled inheritance, there are alternate versions. For each character, each trait, an organism has 2 different versions, or 2 alleles, and that one of those alleles comes from each of the parents. If those 2 alleles differ, then one of the alleles, which he called the dominant allele, is expressed in the hybrid between those 2 parents. The other allele, which is recessive, is not expressed. And that the alleles for each character separate when the plant goes through gamete production through the process of meiosis. From this, he developed his law of segregation. It simply says every trait in each individual is controlled by 2 factors or alleles, and that one allele comes from each parent. They combine in the F1 hybrid individual, but then those alleles segregate through subsequent generations. Then he started looking at crosses that involve plants with 2 or more traits. And when he started looking at 2 traits, or a dihybrid cross, he started looking at one plant that was yellow with round seeds and another plant that was wrinkled with green seeds. And he found that the F1 between that cross gave him all yellow round-seeded plants. And he says, okay, so just like my cross involving a single trait, I get all of the individuals in the F1 to look like the dominant of those traits, the dominant version of those traits. But then when he got to an F2 generation, he ended up with 9/16 that were yellow round, 3/16 yellow wrinkled, 3/16 green and round, and only 1/16 of the progeny were green and wrinkled. So he came up with 2 possible hypotheses for how to explain the inheritance of 2 traits within a cross. And of course, the one hypothesis, which initially would seem to be obvious, is that in fact, when you cross the yellow round and wrinkled green, that you would get the same 3-to-1 ratio down here in the progeny. The fact that he didn't says, okay, these 2 traits do not assort or segregate together. There's independent assortment, so that in fact what you get are yellow round, yellow wrinkled, green round, green wrinkled gametes produced in equal numbers, both for the female and the male gametes, and in fact that explains how you get that 9:3:3:1 segregation ratio. So Mendel says, okay, so there are 2 principles that we can follow in watching traits segregate, or watching traits be inherited. The first is that pairs of genes segregate, the alleles segregate one from the other upon crossing and following generations. The second one is that if you have genes or alleles that control 2 different traits, they segregate independently, so that there is no linkage, and you end up with ratios which basically show independent segregation of two traits. So as I pointed out, not only was Mendel lucky or selective in that all of the traits that he chose to study were either full dominant or recessive, but he was also lucky in that he studied about 7 different traits, and none of those traits were linked together. They all showed independent assortment. Why is that important to a plant breeding program? I mean, this is basic genetics. Why do we care about basic Mendelian genetics in a plant breeding program? I mean, even today, yeah. Well, we'll get to the chromosome segregation in a few minutes, but yeah, that's part of it. But I mean, today's world, we have markers. We can follow genes in detail. Yes?
I was going to say, if the genes aren't linked, then the ratios help you to figure out how many plants you need to grow to get certain amounts.
That's an excellent use application for this. If you're dealing with non-linkage or various degrees of linkage, you get a feel for how big a population you need to work in order to get this independent assortment and recombination. You can predict your phenotypes. And plant breeding basically is the science of either finding variation in genetics, oops, I'm on the wrong slide, finding variation in genetics or creating that variation. And so Mendel's principles of segregation, independent assortment are very valid, and they're principles upon which all of our recombination and selection of different individuals are based. Now, unfortunately, in the real world, none of us deal with 1 or 2 simply inherited traits that show dominant gene expression and no linkage. So what are some of the complications? Well, dominance itself can be variable. Multiple alleles occur at most loci that we studied. There can be lethal alleles, which I won't talk anything more about, that if you have an allele that actually kills the progeny carrying that allele, that messes up your segregation ratios dramatically. But then we have, those are all interactions between alleles at the same gene. In addition to that, we have interactions between genes. Processes such as epistasis and Pleiotropy, which clearly involve genes interacting, controlling traits expressed by other genes. We also have other factors, both genetic and environmental factors. So for the rest of the day's lecture, I'll talk about sort of the top 2/3 of this, and we'll throw in linkage as another complicating factor. And on Wednesday, I'll talk about some of the environmental factors and other genetic interaction factors that can involve this. So when we talk about dominance, Mendel was lucky. All of the traits he studied expressed complete dominance. One parent was purple flowered, the other white flowered. The F1 plants were all purple flowered. You can have cases of incomplete dominance, where the F1 individual looks like something in between the two parents, or more complex, you can have patterns of codominance. In incomplete dominance, both alleles function, both alleles produce functional gene products, and we end up with a phenotype that's different from either of the parents, the homozygous parents. And often we use, instead of a capital letter and a small letter, we use a letter with different numbered alleles, or superscripts. An example of that, if Mendel would have worked with carnations instead of peas, he never would have come up with his laws of segregation and independent assortment. If you cross a red carnation with a white carnation, the F1 generation is pink. So the red gene is incompletely dominant over the white. Therefore, you get a pink expression in all of the F1 individuals. If you follow that to the next generation, you get 1/4 red, 1/2 pink, 1/4 white. So before we go any further with dominance, dominance is not an inherent property of the allele. Dominance describes a relationship between two alleles. So one allele could be dominant to a second allele and recessive to a third allele. And that happens in lots of cases. Or more importantly, more complexly, it could be dominant to a second allele, recessive, codominant, or incompletely dominant to a third allele. I mentioned that we could have multiple alleles controlling a trait. A very simplistic example are the alleles that control blood types in humans. There are 4 possible phenotypes. There's blood type A, which can be controlled. It can either be genotypically AA or AO. It produces A-type antigens or proteins, and it produces antibodies to the B blood type. You could have blood type B, produces B antigens and antibodies to the A blood type. You can have an AB blood type, which produces both A antigens and B antigens and doesn't produce any antibodies. Or you could have an O blood type that doesn't produce either A or B antigens, therefore it produces antibodies to both A and B. You guys know what antigens and antibodies are, right? Antigens are proteins that are foreign to an individual and recognizes an invasive foreign protein, and antibodies is a defense system that basically recognizes and tries to coagulate and tie up those invading proteins. So that basically we say that the O allele for blood type is recessive to both the A and the B alleles, but A and B are codominant. In the blood type AB, both A antigens and B antigens are expressed. There is no dominance or recessiveness between those two. They're codominant. So AB is a universal recipient. It doesn't produce antigens either to the A type or B type. So if you have an AB blood type, you're lucky. You can get a transfusion of blood from anybody. If you're O blood type, you don't produce antigens to either A or B, and so if you're an O blood type, you can give blood to anybody. So a case of multiple alleles involving dominance of A and B to O, and codominance of A to B, and mixtures of expression depending on whether you're A, B, AB, or O blood types. We tend to think that multiple alleles are pretty rare and very special cases. Actually, they're not. Many, if not most, genes have multiple possible alleles. The Rh factor in our blood, the rhesus blood group factor when you're plus or minus, there are at least 18 different alleles involved in expressions of Rh factors. That's why when I was young, people said, well, you get your blood type and you find out if you're Rh plus or minus, and that's all you need to know to worry about potential interactions if you give or receive blood. Well, actually, the Rh factor is a lot more complex than was originally thought. There's one white eye locus in Drosophila that has over 100 different alleles. Now, when you start thinking about, those of you that are working with molecular genetics, when you start thinking about this, it's over 100 alleles that can occupy the same locus, each of which must have slightly different functional capabilities, or we never would have recognized the differential alleles, because all of these were recognized based on phenotypic observations, not based on molecular characterizations. All right, those are interactions between alleles within the same gene. If you have two or more genes acting on a trait, you can come up with what we, for lack of a better term, we call just novel phenotypes. You end up with some sort of interaction between the genes that gives you different segregation ratios than you expect. We end up with epistasis, which is very important. How important do you think epistasis is to plant breeding? Have you had statistics? Have you had any quantitative inheritance? Where basically our friends in statistics, for very valid reasons, say, well, we have all these interactions, but let's just forget about the epistatic interactions because they're so minor that they really don't impact us very much. And so often we say when we're dealing with quantitative inheritance that we have additive genetic factors and dominant genetic factors, and we ignore epistatic factors. Why do you think we do that? Is it because epistatic factors are not important? It's the easy answer. I mean, why don't we spend more time with our quantitative genetic formulas? And we haven't talked yet about molecular genetics, but with molecular genetics, why don't we consider epistasis more than we do?
What exactly is epistasis?
That's the point. What is epistasis? How do you explain it? How is it controlled? We don't know. We don't know. It's a very complex. We do know, and we'll go through some examples, it obviously doesn't involve, probably doesn't involve DNA or RNA interactions between different loci, unless those interactions occur during the synthesis and the folding of proteins. But it obviously involves interactions that occur through metabolic pathways. And so when will we get to understand how important epistatic effects are? I don't know. Maybe if we ultimately develop the science of proteomics to where we are today in the science of genomics, maybe we'll start understanding it. One gene acting on more than one trait, pleiotropy. Do you guys know what pleiotropy is? We'll talk about definitions. All we can say today is all of these are due to metabolic pathways interaction somewhere beyond the gene involving metabolic interactions. So I talked about novel phenotypes. All right, if we look for eye color in Drosophila, you can get a red-eyed and a white-eyed. The F1 are all red-eyed. Simple, right? I mean, a red-eyed individual that breeds true, a white-eyed individual that breeds true. All of the F1 progeny are red-eyed. Isn't this like purple and white pea flowers? Single dominant gene, and the F1 are all red-eyed. So what will the next generation look like? What did the purple and white flowers look like in the next generation? 3 red and 1 white. Well, you get red-eyed and white-eyed in the F2 generation, but you also get brown-eyed and scarlet-eyed Drosophila. And you think, well, that doesn't make sense. That doesn't make sense. The red eye clearly is dominant over the white eye, so we're clearly dealing with a single locus. How do we get, well, it turns out we're not. We get a 9:3:1 ratio, and what we're dealing with basically is one allele, the scarlet allele, that the wild type's red and the recessive is scarlet color. We're dealing with a second allele that the wild type is red, but the recessive controls a brown color. And it turns out that the only way you can get red eyes when you put these two loci together is if you have both a dominant S and a dominant B. Then you get a red-eyed phenotype. Even if you have the dominant S, which by itself controls red, if you have the recessive BB, you get brown eyes. So this is a case where the recessive trait controlled by the B locus is expressed, and the dominant trait controlled by the S locus really doesn't impact that. If you have the recessive SS, you get scarlet eyes, and it doesn't matter what configuration you have at the B locus. And it's only when you have completely recessive scarlet and brown that you get white eyes. I mean, you think about that in terms of the metabolic pathways involved. You think about it long enough, you get a headache. Because it's clearly not a matter of functional alleles versus non-functional alleles. Because here you get brown and here you get scarlet, but you put the two recessives together, you get white. So there's got to be metabolic pathways involved with inhibition of certain factors through metabolic pathways. But again, that's just a case of a novel phenotype that you can get from following through what should be very simple Mendelian ratios. All right, epistatic genes. The effects of one gene override or mask the phenotype of a second. It's not dominant. Epistasis refers to one gene masking the effect of a different gene at a different locus. Dominance refers to one allele masking the effects of a second allele at the same locus. One of the reasons why we tend to say, yeah, but it's kind of like dominance. I mean, it's kind of like being dominant to another gene. So therefore, our dominance effects will cover all of our various epistatic interactions. And maybe they do. If you look at fruit color in summer squash, if you have a genotype that's little a, little b, you get a green squash. If you have a genotype that has at least one dominant B allele, you get a yellow squash. If you have a genotype with at least a dominant A, you get white. But notice, you get white regardless of whether you have dominant B or not. So that when you look for a segregation ratio of a cross between heterozygous AB by heterozygous AB, you get 12 white type, 3 yellow, and 1 green. Saying that as long as you have the dominant A, you're going to get white. In the absence of the dominant factor at the A locus, if you have a dominant B, you get yellow. If not, you get green. So somehow, metabolically, the product of the A gene inhibits the product of the B locus, whether it's dominant or not. But in absence of the dominant A locus, you can see differential expression at the B locus. All right, pleiotropy. A case where one gene controls lots of different traits. And sickle cell anemia is by far the most important example of that. It's one mutant gene, been mapped, it's been characterized. Today, with the human genome being completely mapped, we know in detail what the sickle cell gene looks like. but basically it produces many symptoms. Pain, stroke, leg ulcers, bone damage, jaundice, gallstones, lung damage, kidney damage, eye damage, anemia, and delayed growth. I said that's the biggest or the most well-known example. Is it the only one? No. There are lots of cases of pleiotropy. Phenylketonuria is an enzyme that metabolizes phenylalanine. The dominant codes for a functional, the recessive not for a functional. The primary result, if you can't metabolize phenylalanine in your body, you're mentally retarded. I mean, you have reduced mental capacity, but also your head size, I think, is smaller. Your skin, your hair, your eye color are affected. You have a mousy odor that comes off of your body. You have a peculiar walk, stance, sitting posture. You have eczema and epilepsy, all controlled by presence of the single PKU gene. All right, we'll talk about other factors, incomplete penetrance and variable expressivity. Are you guys familiar with these terms at all? How many have heard about penetrance and expressivity in relation to genes? Good. 2 years ago, I think we had like 2 people in the class that had heard of that. Sex-limited and sex-influent traits, we won't talk about much with plants. And environmental factors, we'll talk about next lecture. All right, incomplete penetrance and variable expressivity. With incomplete penetrance, additional genotypes can show differences in phenotypes. So genetically, 2 individuals are the same, but phenotypically they show differences. And we measure penetrance by saying it's the percent of individuals within a genotype that express the associated phenotype. When we talk about variable, I'll give you some examples of these in a minute. When we talk about variable expressivity, it's the degree to which a genotype is expressed in a phenotype.
So is incomplete penetrance because of the environment? Because we have identical genotypes?
Well, it can be environmentally influenced, but no, we're talking about at single loci. Let me give you some examples and show, typically we say if we're dealing with quantitatively inherited traits, lots of genes that we can expect that the more genes controlling a trait, the more environmental impact you can have. But what we're saying here is for simply inherited traits, we can get variations in expressivity and penetrance. Let's look at eye shape in Drosophila. There's a lobe locus that basically is it's a simple dominant allele, and it reduces the eye size in Drosophila, so that you can have individuals that have the dominant lobe gene and have reduced eye, and individuals without it that have a normal eye. Well, only 75% of flies that have the dominant L allele have the reduced eyes. So 25% of the flies with the single dominant allele have eyes that appear normal. So this is a case I can't quite fathom any environmental effect. I mean, these are fruit flies raised in little bottles in environmentally controlled laboratories. And so there's no obvious environmental impact. It's just this is a gene, the gene that controlled lobed eyes, that doesn't work in all individuals. How can that be? I mean, someday somebody's going to step right up and give me a nice molecular explanation for that. Does it have any relevance in terms of applying genetics to improving plant populations?
Is it similar to electoral laying the eggs and the eggs that are on the top turn into one sex and turn into the bottom?
Well, that has more environmental and developmental implications. No, these are flies that hatch out all at one time. There's a whole generation of new flies, and 3/4 of them, or 75% of them, carry a single dominant gene, but only 50% of them show the trait. So if you do a molecular analysis of those flies, 75% of them carry that gene, but 50% show the reduced eyes, and 25% of that 75%, a third of those guys, show normal eye development.
But it's still correct to say that they have the same genotype. But there might be another gene that affected it. No.
Well, they started with the same genotype, but that doesn't mean that even with the same genotype, that locus necessarily stays in the same position within the genome. And maybe it's equivalent to when we transform plants and we add genes. Have you heard of differences in penetrance of transformed traits? Yeah. I was in industry at the time Monsanto was producing their BT corn. And we worked, as I worked with Cargill, and Cargill and most of the other seed industry worked with Monsanto, looking at lots of different transformational events, some of which showed good penetration of the gene they had inserted, and others didn't show much impact of that gene at all. So maybe it's related to where the gene is located, what it's next to, how it's regulated, but incomplete penetrance is not that rare. Why? Well, actually, a lot of you said you've heard of it. Have you heard of some important examples of it? I haven't. Why? Maybe it's just like epistasis. When we get results like this that we don't understand, we say, well, okay, we get some of these crosses or some of these transformational events we really don't understand, but we're not going to worry about those because we don't understand them. They must not be that important. Let's work with genes that, let's limit ourselves to looking at genes that work. All right, before we run out of time. So the penetrance of the lobe allele is said to be 75%. 75% of individuals that carry the dominant allele show the trait, and 25% of individual flies that carry that allele don't show the trait. All right, let's look at expressivity. This is an autosomal dominant gene in humans that impacts our neural system, as you might tell from the name. Basically, a third of the individual humans that carry this gene don't show any symptoms. Sometimes they show little milk, coffee ole spots, and they have benign skin tumors. But basically, the gene is not very well expressed in a third of the individuals with it. A third don't have any symptoms for most of their lives. But they have episodes where they really get sick, and they can get very, very sick to the point of near death. A third of the individuals that carry this single autosomal dominant allele are severely affected and symptoms severe enough that it can cause premature death. All right? So, single dominant gene, individuals that carry it show different degrees of expression of the gene. All right, we're going to go through next lecture, environmental effects. We'll talk about that. I want to get to some little bit on Morgan, which hopefully you all guys know about genes now that they occur on chromosomes and always have. Even the genes and organelles are on somewhat our mitochondrial and chloroplast chromosomes. Linked genes are on the same chromosome, and genes can be exchanged between chromosomes, and that exchange occurs during meiosis. And the closer genes are located on chromosomes, the less likely you're going to get a crossover between them. This is just a deal, you can work through that. It just shows they're on chromosomes, therefore you can have two different arrangements of those genes, and we'll go through meiosis in a later class just to review some of the things. So, you know, in essence, when Mendel came out to independent assortment, if in fact you have full linkage, you do get dependent assortment. Crossing over. I'm hoping you guys know most of this so I can rush through it. If you have questions, stop me. Crossing over occurs when the sister chromatids pair and the homologous chromosomes line up and pair together, and so you get exchange of genetic material. Occurs during meiosis. Well, the closer two traits genes are, the less recombination. The farther apart, the more. In fact, the percent recombination is used as a measure of genetic distance. This is before our molecular terms where we actually measure genetic difference. So that basically if we have two traits, we have gray eyes or gray flies with normal wings versus black with reduced wings, and we take a heterozygous individual and cross it against a homozygous recessive individual, we should get a 1-to-1 ratio of these two traits that are linked. And if we get any of the gray with the reduced wings or the black with normal wings, it's due to recombination between those two genes. And so in this case, we came out with most of our offspring parental type. 391 total were recombinant types. So we had 2,300 total offspring. 391 showed recombination. That's 17% recombination, or 17 map units between those two genes. Pretty much review, I hope, on mapping. You can run the mapping. You can do a chi-square analysis to determine whether what you're observing is statistically valid or whether you're observing some sort of environmental impact that's skewing your ratios. All right? So basically, on what we've talked about so far, the reason why all of this genetic is sort of interest to me to plant breeding is plant breeders are looking for genetic variation. So how some of the ways that genetic variation can originate? Well, number one, this independent assortment. I mean, if you're dealing with a lot of genes in organisms with lots of chromosomes, and I mean, you know, we deal with maize, which I bred, and we only have 10 pairs of chromosomes. But, you know, when you deal with sweet potatoes with 80 pairs of chromosomes, you can begin to see that the amount of reshuffling of genes due to the fact that just genes on different chromosomes are assorting independently can be tremendous. Well, for most of our crops, that doesn't give us a lot of genetic variation. Crossover occurring between different genes and getting new recombinations. That happens very, very frequently. Every time our crops go through a sexual cycle, we can get significant recombinations. When these chromosomes pair, we get significant amount of crossing over. And probably in the development of our crops, this genetic recombination through crossover has led to more new genetic variation than any of these other factors. When we're talking about plants in populations, whether plant breeders are making the crosses or nature's allowed to control the crosses, random fertilization. We can get outcrosses of material coming in, and we can get genetic variation. And then finally, mutations can give us genetic variation. I'll tell you later, most mutations are lethal or harmful, and you don't want to mess with them, but some are not, and they're spontaneous, and they can occur at relatively reasonable rates, and they can be reversible. But to give you the bottom line, mutations probably account for a very small part of the genetic variation within crops that we deal with today. Independent assortment probably accounts for a larger proportion of genetic variation in a cross between two distinct individuals. Recombination probably accounts for the majority of the new recombinants of genes, or the new individuals with genetic variation. So there are several references there. I went through those a little quickly that sort of give you reviews of genetic, and a lot of these will give you little graphics and animations, and if you're rusty on your genetics, review it. Otherwise, are there quick questions? All right.